Hello everyone, my name is Akashdeep Singh, and today I will be talking about stem cells. Stem cells have a great promise in future medication treatments with so many possibilities. Today you'll learn what a stem cell is, the types of stem cells, and the current uses of stem cells, the state of research that's been going on as of right now, and many more other things. You might have heard about stem cells in the news, and perhaps you might wonder if they will help your loved ones or you if with a serious disease. You may wonder that what stem cells are, how they're being used to treat diseases and injuries, and why they are a subject of such a vigorous debate. What are stem cells? Basically, stem cells are cells that has the ability to continue to divide and differentiate into various other kinds of cells and tissues. Self-renewal and multipotency. What does it mean to self-renewal? Self-renewal is the ability to generate daughter cells identical in phenotype and potence to parental cells. Maintenance of streamlinedness and homeostasis of stem cell number and cytochronological functions. What does it mean to multipotency? Basically, is the ability of a stem cell to differentiate into different cell lineages, including mes, endo, and ectoderm. Cells that can cells can self-renew, duplicate, and differentiate. What does it mean to self-renewal? Basically, it means maintain the stem cell pool. One cell can give rise to others. Why differentiate? Differentiate. One stem cell can give rise to many specialized cells, repair and replenish damaged cells throughout the lifespan. Why is there such a big interest in stem cells? The first thing is that they increase our understanding how diseases happen or occur. So, how they increase our understanding is that by watching stem cells mature in cells, in bones, heart muscle, nerves, and other organs and tissues, the researchers and doctors can observe these and understand how diseases and conditions develop. The second thing is that stem cells they generate healthy cells and they replace the diseased ones in our body. Stem cells basically how it works is that we have a disease. You inject the stem cell in your body. When the stem cells go inside your body, they replace the ones that are damaged or that has the diseased ones, and they act as those new cells in that type of muscle or what type of disease you have. For example, if you have a heart muscle, it will replace those diseased heart muscles or injured ones with new ones in order to work. That's what it means. Third, stem cells, they help us test new drugs for safety and effectiveness. Before using investigational drugs in people, research can be used for some type of stem cells to test the drug for safety and quality. This type of testing is mostly likely to have a direct impact on the drug development, first for the cardiac toxic testing. New areas of study include the effectiveness of using human stem cells that have been programmed into the tissue-specific cells to develop new drugs. For the testing of new drugs to be accurate, the cells must be programmed to acquire properties of the stem cell target by this drug. Techniques to program cells into specific cells continue to be studied at this point. For instance, nerve cells could be generated to test a new drug for a nerve disease. Tests could show whatever the new drug has affected on the cell or wherever the cells were harmed during the process. So I'll be talking about the type of stem cells that we have. The first one is embryonic cells. Basically, embryonic cells, they come from the embryos that are about 3 to 5 days old. And when embryo is in this stage, it's called a blastocyst, which has about 150 cells. To add on that, embryonic stem cells, they include porphyrins, which cells can form any cell type, and they're in the blastocyst. Let's talk about the day 5 of the embryo. They undergo differentiation, stop dividing. Cells are topiant and can differentiate into any cell. After the first stage of differentiation, they are no longer topiant. The second stem cell I want to discuss is adult stem cells. They're found in small numbers in most adult tissues, such as bone marrow or fat. And adult stem cells, they include multipotenants, and multipotent basically is cells that can differentiate but can form a number of other tissues located in the fetal tissue, cord blood. And adult stem cells, they're self-renewal, they divide rarely, and they have high potency, and they divide rapidly. The last stem cell I'm talking about is induced pluripotent stem cell. Scientists have successfully transformed regular adult cells into stem cells using genetic reprogramming. By altering the genes in the adult cells, researchers can reprogram the cells to act similarly to embryonic stem cell, where it's basically similar as the embryonic stem cell, where the embryonic stem cell it contains a pluripotent, where this includes an induced pluripotent. So the difference between the embryonic cell and the adult cell, the difference is that embryonic is indefinite self-renewal, adult is limited self-renewal. Embryonic it differs to all cell types in organism, pluripotent, meaning ecto, mes, endo. Adult, the difference into restricted cell type multipoint, so they cannot differ into ecto, mes, or endo where embryonic can. Embryonic it changes upon stimulation, where on the other hand, adult they emphasize the ability to need to activate by clues. Example, injuries. And you can see how in these diagrams, how I talked about the 5 days, the potent cells, and this is the blastoid. Over here, the adult stem cells, how it they're usually located in the bone marrow and all liver, all this affected, all these factors. So the next topic I want to talk about is stem therapy. So basically, therapy is pretty important because it actually helps people that are injured or diseased, and these are the 6 stages of stem therapy. So basically, for example, you have a heart muscle, like something's wrong with your heart muscle inside of your body. Basically, these stem cells they will be injected into your body, and once they enter the body, they will replace those muscle cells that were diseased or injured, like I said previously in the other slide, and they'll act as that new muscle cell. Basically, which is pretty good. How you can see, like, look right here. You can see how stem cells in this diagram, you can see how it self-replicates and differentiates into multiple tissues, bone, muscle, cartilage, and fat. And there's other things that happen, which is pretty good. But there's still research being done on stem therapy where it's not 100% accurate, but so far the results have been pretty good. So the problem using embryonic stem cells in humans is that for embryonic cells to be useful in research, I mean in people, research must be certain that stem cells will be differentiated into specific cell type desired, and research has been discovered to directly stem cells to be specifically types of cells, such as direct embryonic stem cells to become heart cells, and research is still going on. Embryonic stem cells are also grown regularly or specific into different cell types spontaneously. Research are studying how to control the growth and the differential embryonic stem cells. Embryonic stem cells might also trigger an immune response in which the body might attack the stem cells as a foreign invaders, or the stem cells might fail to function normally, which is a known consequence. Research, like I said, is still going on to avoid these problems where stem cells are getting attacked or many other problems that might occur. The last thing I want to talk about is theoretical cloning. I'll touch light on this, but theoretical cloning is something called the somatic cell nucleus transfer. It's a technique to create verticalized stem cells, independent verticalized eggs. In this technique, the nucleus, which contains the genetic material, is removed from the union of fertilized egg, and the nucleus is also removed from the cell donor. Where the cell donor nucleus is injected in the cell, replacing the nucleus was removed. In this process, it's called nuclear transfer. This allows the egg to divide into form blastocysts. And that's basically what cloning is. You can talk here. I talked about right here nuclear transferring, as you see in the diagram. There's a huge cycle that goes on. Embryonic stem cells are used in this cloning. It's successful as of right now. It's not. Basically, half of this information, most of the stuff has been successful, and most of it hasn't. And research is still being done. And thank you for listening to me, and have a good day. Thank you.